Hello friend, welcome to all of you for the session Respiration in Plant session 4. Myself BP Shah, PGT Bio, Kendriya Vidyalaya Kesapuram. Today in this session I will discuss the left one which I have discussed in my last session up to the glycolysis. After the glycolysis, the next process in aerobic respiration, that is Krebs cycle. Before we going to that, first of all the question I have asked in my last session, now I think you all are able to answer these questions. First of all, what are the questions that think over? My first question was, what are the differences between the glycolysis and fermentation? The second one, aerobic respiration and fermentation. Third one, aerobic respiration and anaerobic respiration. Next one question, what are the end product of fermentation in yeast? And last question, name the two instances where lactic acid is formed by fermentation. Just we have already discussed fermentation. In another way it is called anaerobic respiration. The product, what will be the product is formed. My first question is, what are the differences between glycolysis and fermentation? The answer is, glycolysis occurs during the aerobic and anaerobic respiration in both cases. In both cases, glycolysis is the common way by which the process is going on, and the fermentation is a type of anaerobic respiration. Next one differences is pyruvic acid produced at the end product of the glycolysis, whereas the end product of fermentation is ethyl alcohol, that is called ethanol, and lactic acid. My second question is, what are the differences between aerobic and anaerobic respiration and fermentation? Aerobic respiration, as the term indicate, oxygen is used. In fermentation, it occurs in absence of oxygen. In aerobic respiration, it occurs in both cytoplasm, where the glycolysis takes place, and mitochondria, where Krebs cycle and oxidative phosphorylation electron transport chain takes place. Fermentation only takes place in cytoplasm. The result of aerobic respiration is carbon dioxide, water. Result of fermentation is ethanol or lactic acid. It depends upon the where it occurs, either in muscle cell or in yeast or in bacteria. The aerobic respiration output, that is 36 ATP is produced or 38 ATP. Here in case of the fermentation, only 2 ATP is produced. Now, my next question was, what are the differences between aerobic and anaerobic respiration? Same thing is here. Anaerobic respiration is also called fermentation, same thing, either anaerobic respiration or fermentation is the same way. It takes place, aerobic oxygen is used here in absence of oxygen in case of anaerobic. Aerobic involved in cytoplasm and mitochondria. Only cytoplasm will take part in the anaerobic respiration. End product carbon dioxide, here the end product is lactic acid and ethanol. 36 ATP is produced, 2 ATP is produced. Now, the next question is, what is the end product of fermentation in yeast? There are two products in fermentation that we have already discussed in detail in my last session, that is, in yeast ethyl alcohol is formed, or in case of muscle cell lactic acid is formed. So, here is the answer is the end product fermentation of yeast is ethyl alcohol, that is also called ethanol. The last question was, name two instances where lactic acid is formed by fermentation. First of all, the answer is during fermentation by lactic acid bacteria, which involved in the conversion of milk into curd. Or second one, two instance, the second one instance is during strenuous exercise or vigorous exercise, when there is a no sufficient of oxygen is obtained by the organism. So, the static muscles accumulate the lactic acid. So, these are the two instances where the lactic acid is formed. One is in the formation of curd and second one is vigorous exercise, where the lactic acid are accumulated inside the muscular cell or the static muscles. Now, before we going on the Krebs cycle takes place in aerobic respiration, where the glucose molecule is broken down in presence of oxygen, which I have already discussed, and the product will be carbon dioxide, water and energy. In this process, in oxidative carboxylation and glycolysis, there is no involvement of oxygen, but in case of Krebs cycle, the molecule of oxygen is used, which is the ultimate acceptor of the proton and electron removed from the substrate. It is carried by enzyme of the matrix and the inner which are present in the inner membrane of the mitochondria. The aerobic respiration have two distinct mutually process, which are already told, Krebs cycle and terminal oxidation. It is also already discussed, before participating in TCA cycle, pyruvic acid first of all converted into acetyl coenzyme, and during this process NAD is reduced into the NADH plus H plus and carbon dioxide is released. The acetyl coenzyme now, which is the product of oxidative decarboxylation, it enter into the Krebs cycle for further degradation. Now, the various steps of the Krebs cycle. In eukaryote, it operates in the matrix of the mitochondria. The substrate of Krebs cycle is acetyl coenzyme, and the acceptor of acetyl coenzyme in mitochondrial matrix, that is OAA, that is called oxaloacetic acid. We have already discussed. These are the different steps of the Krebs cycle. First, condensation, second one isomerization, third one dehydrogenation, fourth one decarboxylation, fifth one oxidative decarboxylation, sixth one substrate level oxidation, seventh one dehydrogenation, eighth one hydration and ninth one dehydrogenation. After the dehydrogenation, the product will be formed, that is called oxaloacetic acid. Now, one by one these steps, how this step is going on. The first of all, the condensation process. The first step of condensation, it takes place in mitochondria, and the first product which is formed in the oxidative decarboxylation, that is acetyl coenzyme. This acetyl coenzyme which is 2 carbon compound, that is called acetyl coenzyme. We have already discussed this acetyl coenzyme acts as a substrate of the Krebs cycle. It combined with oxaloacetic acid this oxaloacetic acid, the structure are of this type COH, CH2, C double bond O, it is here the ketone group is there, 4 carbon compound, here is the 2 carbon compound. The acetyl coenzyme combined with the oxaloacetic acid, this is the first reaction, that reaction is called condensation. So, this is the first steps of the Krebs cycle, that is condensation. This acetyl coenzyme and oxaloacetic acid, it is going to be combined in presence of enzyme, that is called citrate synthetase. And it forms citric acid, which is 6 carbon compound, COH, CH2, COOH. Here 3 acid group are there, COH group 1, 2, 3, 3 carboxylic acid is there. So, in case citric acid or TCA, tricarboxylic acid cycle is called. So, Krebs cycle in another way it is called citric acid, because first product after the condensation it is formed, that is citric acid. This citric acid contain 3 carboxylic group. So, this cycle is called TCA cycle. Now, this oxaloacetic acid, it is combined with coenzyme HSCOA. Now, the next reaction is isomerization. In isomerization what happened? This citric acid is converted into cis-aconitic acid, and cis-aconitic acid again converted into isocitric acid. At the time of conversion of citric acid into the cis-aconitic acid, the water is released. So, this process again it is called the sub step of the isomerization, that is called dehydration, where citric acid is converted into cis-aconitic acid and the enzyme here involved is aconitase. After that, this cis-aconitic acid is converted into isocitric acid. It also require water molecule. So, this step is called rehydration. Rehydration. Here the citric acid is converted into isocitric acid, that the same formula is there. So, this reaction, the second step is called isomerization. So, in isomerization the citric acid first of all converted into isocitric acid and the isocitric acid converted into cis-aconitic acid and cis-aconitic acid converted into isocitric acid. Now, the structure is this type COH, CH2, COOH, CH2, COOH. Here there is the removal of water, as we have already. From where this water is going to be removed? This OH and this H is going to be removed. So, you can easily understand from where the water molecule, 2 hydrogen and oxygen is released. So, you can easily write the next step, that is cis-aconic acid without any problem, that is remove this group, the red one, COOH, CH2, COOH, CH, COOH. This is the structure of cis-aconic acid, but it is not true. When you write the structure formula of any organic compound, you must know that the valency of carbon must be 4. Here in this cases the valency of carbon is 1, 2, 3. So, this reaction is not sufficient. Here the single bond is converted into the double bond. Now, the structure is cis-aconic acid. Now, this process is called hydration, because water is removed and the enzyme is aconitase. What happen in next reaction? We have already discussed. The third one process of the isomerization, that is the rehydration. Rehydration means adding of water. Water molecule is going to be added. This water molecule is going to be added and the formula will be, the product will isocitric acid. Now, we can easily write the isocitric acid formula. This 2H is used, H2O is going to be used here, COOH, CH2, CH, COOH, CH, OH, COOH. What is the difference? This H, OH is here and H is here. So, you can easily write where the H and OH is used, we can easily write the structure, that is isocitric acid. So, this H is used. The Krebs cycle, the first step, that is called condensation, which I have already discussed. In condensation process, the acyl coenzyme combined with the oxaloacetic acid and it form citric acid. Now, the second process is called isomerization. In this isomerization, two processes is involved, that is called dehydration and rehydration. In dehydration process, there will be the removal of water, and rehydration process, there is the addition of water. In dehydration process, the citric acid, which is the condensation product, it is, the structure is this, COOH, CH2 and COOH. Now, from the citric acid, water molecule is removed. So, this process is called dehydration. D means removal of water molecule. So, water molecule is going to be removed. This water molecule is removed from citric acid, you know easily from this group 1H and OH, 1H and OH is removed from this carbon and this carbon, and the process is called dehydration. The enzyme involved is called aconitase. So, this process is called dehydration process. Now, the structure you can easily write. See, after removing the water from the citric acid, now the rest one thing is COOH. Here CH is only remains and COOH. Now, this is called cis aconitic acid. But this structure is not correct, because this carbon has only 3 valency. So, one double bond is required. Now, this reaction, this structure is complete. Now, after the cis aconitic acid, it undergo rehydration. Rehydration means adding of water. This water molecule is going to be added to the cis aconitic acid, and the enzyme involved is the same, that is aconitase. Now, this water molecule after addition it form isocitric acid. Isocitric acid. This water molecule is going to be added here. Now, the structure will be COH, CH2, CH, COOH, CH, OH and COOH. Now, you will easily understand this OH have H and OH. This H added here and this OH added here. So, you can easily write the formula of isocitric acid from cis aconitic acid, that is, now the entire reaction from the citric acid to the isocitric acid, there is the same structure formula. So, this entire reaction is called isomerase, but 2 steps are there. One step is called dehydration, another step is called rehydration. Now, the isocitric acid now converted into oxalosuccinic acid. Oxalosuccinic acid. Here the removal of hydrogen. So, see this reaction how it is going on. The from the isocitric acid COOH, CH2, CHCOOH, CHOH, COOH, isocitric acid. It is converted into oxalosuccinic acid. As we have discussed, here is the removal of 2 hydrogen. These 2 hydrogen removed from here, this carbon. So, you can easily write the formula of oxalosuccinic acid, that is COOH, CH2, CH, COOH, C, you know that oxygen have double bond, COOH. So, without any problem you may write the formula of oxalosuccinic acid by knowing from where this 2 hydrogen is going to be removed. So, removal of hydrogen from the isocytric acid, this is called dehydrogenase. The enzyme here involved is dehydrogenase. This enzyme involved in isocytric acid. So, we can say when we add substrate isocytric dehydrogenase. Now, what is the fate of this hydrogen? This hydrogen is accepted by a coenzyme which you have already familiar, that is NAD plus. It converted into NAD H plus H plus. Now, this coenzyme nicotinamide adenine dinucleotide, it is going to be reduced and form NADH and H plus. Now, this will be further used in the electron transport chain and the oxidative phosphorylation, where reoxidation of this reduced coenzyme is going on and it helps in the formation of ATP. Now, the oxalosuccinic acid, this is the dehydrogenation process, this one is the third step of the Krebs cycle. Now, we are going to the fourth step of the Krebs cycle, that is decarboxylation. As the term indicate, from the oxalosuccinic acid the carbon dioxide is removed. This carbon dioxide is removed. So, the enzyme, nothing to worry, decarboxylase. Here the substrate is oxalosuccinate. So, we can name it oxalosuccinate decarboxylase enzyme is used and the carbon dioxide is removed. When we know this carbon dioxide removed from where of the oxalosuccinic acid, we can easily write the next one step, that is from the oxalosuccinic acid after the removal of carbon dioxide, there is the formation of alpha ketoglutric acid, which is the intermediate product of the Krebs cycle, which is formed by the beside the carbohydrate other substrate also form the alpha ketoglutric acid. This carbon dioxide is removed from here. So, we can easily write the formula of alpha ketoglutric acid without any problem, that is COOH, CH2, here CH2 after the removal of carbon dioxide H and H molecule are there. So, we can easily write CH2, here COOH is there, sorry, here this one again write C double bond O and COOH. So, these are very important molecule, you can see in amide synthesis, that is alpha ketoglutric acid is formed after the removing of carbon dioxide from the oxalosuccinic acid. This alpha ketoglutric acid, now here the substrate level phosphorylation takes place and there is the formation of it undergoes the substrate level. In the next step, this alpha ketoglutic acid, first of all, here the 2 hydrogen molecule is removed, and in that cases carbon dioxide is also removed, and the product will be succinyl coenzyme, succinyl coenzyme A. This hydrogen molecule, this hydrogen is removed, and this hydrogen is accepted by NAD plus and is again converted into NADH plus H plus, and the product is succinyl coenzyme, and we are going to here add HSCOA as the precursor of succinyl coenzyme. Here the structure will be COOH, CH2, CH2. This carbon dioxide is removed from here, and this hydrogen is removed from here, and this one from the succinyl coenzyme. This hydrogen is removed, this carbon dioxide from here. So, we can easily understand the formula of succinyl coenzyme. So, only CO is here and SCOA. So, this structure is called succinyl coenzyme, and here two things is going to be happened, decarboxylation as well as dehydrogenation. So, the enzyme involved here is the enzyme here decarboxylase oxalosuccinic succinyl decarboxylase. Succinyl decarboxylase is involved, and the alpha ketoglutic acid converted into succinyl coenzyme, and the product will be the there is another NADPH2 is formed, NADH plus H plus. Now, the succinyl coenzyme is undergo substrate level ATP synthesis, that is COOH, CH2, CH2, COSCOA. This is succinyl coenzyme. Now, it undergo substrate level oxidation. The enzyme here used as succinate thiokinase, and the hydrogen is removed, and here GDP converted into GDP plus PI, it converted into GTP plus ADP, which help in the formation of ATP. Here ATP is formed from the substrate directly. So, this reaction is called substrate level phosphorylation. This substrate level phosphorylation also takes place in photosynthesis in non-cyclic phosphorylation. Now, the succinyl coenzyme now converted into succinic acid. The formula of succinic acid COOH, CH2, CH2, COOH. This is the formula of succinic acid. Now, from the succinic acid, there is the formation of fumaric acid. There is the formation of fumaric acid. This is the 6th level reaction, that is called substrate level oxidation from succinyl coenzyme to the succinic acid. Now, the Next and the seventh step of the Krebs cycle, that is dehydrogenation. In dehydrogenation, hydrogen molecule is removed. This hydrogen removed from this and this. The removal of hydrogen, the enzyme involved is dehydrogenase, no doubt. Here the substrate is succinic acid, so we can say succinic dehydrogenase. Here one thing is very important, from the succinic acid to the fumaric acid, this hydrogen is released, this is not accepted by NADP, NAD. So, this one is accepted by FAD, flavin adenine dinucleotide, and it converted into FADH2, which undergo terminal oxidation or electron transport chain, and after the reoxidation of this FADH2, it gives 2 ATP, which we will see later on. Now, the structure of fumaric acid we can easily write by removing this one hydrogen from here, one hydrogen from here. So, we can easily write the formula of COOH here. After the removing of one H, only H is there. Here after removing H, only CH is there, COOH is there. Now, the structure is not complete because the carbon have no fold valency. So, here is the double bond in between carbon carbon atom. So, now it is the formation of fumaric acid, there is the dehydrogenation process takes place. So, this one is the seventh step of the Krebs cycle, that is called dehydrogenation. Now, from the fumaric acid it converted into malic acid. At this time conversion of fumaric acid to malic acid, there is the addition of water, water is going to be added. So, the reaction or the structure you will find that this is the structure of fumaric acid, COOH, CH, double bond here, CH, COOH, that is fumaric acid. This fumaric acid, after the addition of water molecule, it converted into malic acid. Where this water molecule is going to be added? The enzyme involved here, that is fumarase. From water, one hydrogen is going to be attached here and another OH is going to be attached here. So, you can easily write the formula of chemical structure of fumaric acid, COOH, as we have already told. This H combined with this H, it form CH2. Now, one H is already here. Now, the OH is here and COH is already present. So, this structure is called malic acid and the enzyme involved here is fumarase. Now, this process is called hydration. Now, the last step of the Krebs cycle, from the malic acid, the acceptor of acyl coenzyme, that is oxaloacetic acid is formed. So, from the malic acid, oxaloacetic acid is formed. In this process, there is the removal of hydrogen from the malic acid to the oxaloacetic acid. This hydrogen again it is accepted by coenzyme NAD plus and it converted into NADH plus H plus. Now, the product is oxaloacetic acid. Now, where this hydrogen is going to be added? This hydrogen is added to the NAD and it converted to NADH2 and removal of this hydrogen from here. This hydrogen and this hydrogen going to be removed from the malic acid. So, we can easily write the structure of oxaloacetic acid, which already in my first reaction CH2, here only oxygen is present. So, oxygen is written as double bond and COOH is there. So, there is the formation of oxaloacetic acid and removal of hydrogen from the malic acid. The enzyme is used dehydrogenase. Here the substrate is malic acid. So, we can easily say malic dehydrogenase enzyme help in the conversion of malic acid into the oxaloacetic acid. Now, we are started from the oxaloacetic acid which combined with acetyl coenzyme and again this reaction for after the 9 successive step oxaloacetic acid is again formed. So, the entire reaction are in the cyclic form. So, this cycle is called Krebs cycle. Now, we can overlook this reaction is as now the acetyl coenzyme here combined with the oxaloacetic acid in presence of citrate synthetase, citric acid is formed. Citric acid converted into 6-aconitic acid. This 6-aconic acid converted into isocitric acid. This isocitric acid converted into oxalosuccinic acid. Oxalosuccinic acid form alpha ketoglutric acid. Alpha ketoglutric acid after the removal of hydrogen and carbon dioxide, this forms succinyl coenzyme. Here the substrate level formation of ATP takes place, substrate phosphorylation takes place and this succinyl coenzyme converted into succinic acid and GDP and inorganic phosphate finally form the ATP. From the succinic acid fumaric acid is formed. Fumaric acid the converted into the malic acid and malic acid converted into oxaloacetic acid. So, the entire Krebs cycle when we have a look, there is the formation of NADPH2 here. Here one NADPH2 is formed. First NADPH2 is formed from the isocitric acid to oxalosuccinic acid. So, when we summarize the steps of where the ATP is formed, we have already discussed in ATP formation, in glycolysis glucose is converted into glucose 6 phosphate, 1 ATP is consumed. Fructose 6 phosphate when converted into fructose 1,6 bis phosphate, 1 ATP is consumed. At the time of conversion of 3 phosphoglyceraldehyde, 1,3 bis phosphoglyceric acid, 1 NADH2 is used. So, NADH2 first NADH2 is used in case of glycolysis at the time of conversion of phosphoglyceraldehyde when converted into 1,3 diphosphoglyceric acid. So, this one is the first NADPH2. The second NADPH2 we have already discussed, that is in oxidative carboxylation when pyruvic acid converted into acetyl coenzyme, one NADPH2 is formed, NADH2 is formed, which release ATP. Now, in Krebs cycle we have seen the first NADH2 is formed isocitric acid when converted into oxalosuccinic acid, and second one alpha ketoglutric acid converted into succinyl coenzyme, and third one when malic acid is converted into oxaloacetic acid. These three, the 7th one, 8th one and 11th one hydrogen is released and accepted by NAD plus and converted into NADH2. The third one, the 10th one step, where the conversion of succinic acid into the fumaric acid, this hydrogen is accepted by FAD and converted into FADH2, and one direct level of ATP is synthesized, that is succinyl coenzyme converted into succinic acid, where GDP combined with the inorganic phosphate, GTP is formed, then ADP is formed, this again converted into ATP. So, this is the one look where the NAD plus converted into NADH2 and FAD is converted into FADH2, which are going to be used in the further reaction of that electron transport chain and so on. This is the look. Now, what are the significance of Krebs cycle? The Krebs cycle is the common pathway of carbohydrate, protein and fat. We can summarize this thing in the same way. The glucose molecule, which is the substrate, it converted into firstly pyruvic acid converted into acetyl coenzyme. Now, we are able to say this acetyl coenzyme enter into the tricarboxylic acid cycle, that is the Krebs cycle, which is accepted by oxaloacetic acid and it converted into citrate. This citrate again converted into alpha ketoglutric acid, which we have already discussed in details. Now, this alpha ketoglutric acid form succinate and this succinate again return back to the oxaloacetic acid. This alpha ketoglutric acid help in the formation of glutamate and this glutamate help in the formation of protein, and oxaloacetic acid also help in the formation of aspartate, and from the aspartate protein is formed. So, and the fatty acid is converted into fatty acid, fat is converted into fatty acid, and this fatty acid directly involved in the acetyl coenzyme formation. So, we can use in significance of the Krebs cycle. In Krebs cycle, there is a common metabolic pathway for carbohydrate that we have seen. The glucose is directly involved in the Krebs cycle. Fatty acid, which is directly involved in the formation of acetyl coenzyme. This acetyl coenzyme directly involved in the tricarboxylic acid cycle, and the protein help in the formation, it vice versa, formation of aspartate. Aspartate again formation is the oxaloacetic acid. This oxaloacetic acid combined with the acetyl coenzyme, it form citrate. This protein converted into glutamic acid, this glutamic acid converted into alpha ketoglutric acid, which directly involved in the TCA cycle. So, we can say it is a common metabolic pathway for the carbohydrate, fat and protein. The next one significance, which is very important, as we have discussed, the ATP is the energy currency. Major sources of ATP molecules, which is stored in the form of NADH plus and FADH2. We have already seen this NADH and H plus is formed in the various step of the Krebs cycle, and FADH2 is also formed. Both are the coenzymes. This coenzyme is going to be reduced and it is going to further oxidize and release ATP molecules. This NADH2, NADH plus H plus, it releases 3 ATP, and FADH2 it releases 2 ATP, which we have to see in the next session. It is the immediate act, intermediate acts as a source of synthesis of other molecules. Its intermediate like succinyl coenzymes, this used for the synthesis of chlorophyll. The succinyl coenzyme, which is the product of the TCA cycle, succinate. Before formation of succinic acid, there is the succinyl coenzyme which we have already discussed. This succinyl coenzyme, this one succinyl coenzyme, this step is directly involved in the synthesis of chlorophyll molecule. We know the chlorophyll molecule is very important, which help in the photosynthesis. In my last lecture of the photosynthesis, the importance of chlorophyll, it receives particular wavelength of light from the radiant energy, and it is able to convert the radiant energy into the chemical energy. The next intermediate product like OAA, oxaloacetic acid, and alpha ketoglutric acid, it is already seen. Oxaloacetic acid and alpha ketoglutric acid, which is the intermediate product of tricarboxylic acid cycle, it help in the synthesis of amino acid. So, we can summarize the entire reaction with the help of these things. It is the major step where the carbohydrate, fat and protein involved. Now, we are able to think this question also. First thing is, what are the main steps of aerobic respiration and where it takes place. So, you can easily think this question, the aerobic respiration, here is the involvement of oxygen, and there are the many steps like we have already discussed glycolysis, oxidative decarboxylation and Krebs cycle and terminal oxidation takes place. The second question is, which enzyme is required for the synthesis of ATP? We know that the various product of ATP is formed directly or indirectly. Indirect for the substrate level phosphorylation takes place in Krebs cycle as well as in glycolysis, ATP is synthesized. For the synthesis of ATP, it requires particular substrate like ADP and PI, and the enzyme involved in these cases are ATPase, which are found in the mitochondrial matrix, that is called oxysome in F1 particles. And now, think over this questions, which type of respiration result in the formation of water? The ultimate product is water, as we have already discussed in my first class. In respiration what happened? The carbohydrate molecule, it either combined with oxygen or in absence of oxygen. When it combined with oxygen, the result will be carbon dioxide and the water. This water is the ultimate source of the when oxygen is present. So, which type of respiration result in the formation of water? From this reaction you have easily answered it. Now, next thing is where this NADP and FADH2 is going on. Now, the next step, this NADH2 or FADH2, H plus and FADH2, it is going to be reoxidized. After the reoxidation, this results formation of ATP. Here the reduced coenzyme is converted into ATP. So, this type of phosphorylation is called oxidative phosphorylation. So, we link that this oxidative phosphorylation is the next step of the aerobic respiration. It is also called terminal oxidation. So, it is better to say the oxidative phosphorylation to the terminal oxidation, because it is the final step of aerobic respiration which involves release of proton and electron from the reduced coenzyme and their ultimate reaction with the oxygen. This proton is combined with oxygen in this way. The electron 2H plus, this proton combined with half molecule of oxygen and it converted into the water. It takes place at the end point of the respiration. So, this one is called terminal oxidation. So, oxidation takes place at the end point, ultimate reaction with the oxygen, and there is the formation of water. It is known as terminal oxidation because it occurs at the end of metabolic reaction of the aerobic respiration. Now, this entire reaction which is formed, that is NADH plus H plus, is converted into NAD plus 2H plus and electron. The FADH2 is returned back to FAD 2H plus and 2 electron. Finally, this reaction combined to form the water. This will be terminal oxidation, which followed by two step of reaction, that is electron transport chain and the oxidative phosphorylation, which will be discussed in the later classes. Now, think over this, which will be you are able to now answer this. What is the main step of aerobic respiration, where it does takes place? Which enzyme is required for the synthesis of ATP? Which type of respiration result in the formation of water? Besides this, various types of question, what is substrate level of phosphorylation, oxidative phosphorylation, photophosphorylation takes place. So, this will be discussed in my next class. Thank you.